clicker pointer?
I'm just going to use the screen.
Cool. Again.
How much lighting do you like, Jonathan?
Not too dark.
Okay.
People will be snoozing.
Let's see. Just for the screen. That one. Maybe we do that. Yeah.
Oh yeah, that's good.
That's good.
Is that all right? Okay. So, there we go. And did Jessa ask you about having lunch with the grad students, if that would be a possibility?
Yeah, I said basically yes. I have something I have to run to after the seminar, but if they tell me where to meet,
Okay.
Yeah, it's in 176 Hutchinson,
Yeah, fine.
right by the workshop.
That's easy.
Hey, Tom.
Hey, how are you?
Good.
Good.
How's it going?
All right.
That's the typical Monday plan. Yeah, is noon in 176. Yeah, Tyler. Nice to actually formally meet you.
Good voice.
Yeah.
And so, that would be great. Noon, please, ready to
In 176.
Yeah, sure.
Right now.
I'm around.
maybe,
Sure, absolutely. Yeah, it typically goes to Greg since he's the president, and if he's not there, I have to stay. So yeah, someone will definitely do that. Yeah. In fact, I'm part of that planning too. Yeah. So we got one sandwiches and soda and stuff like that. Yeah, it should be good for us. I'm privileged to be your grad student. Tyler. Yeah, and I'm one of the. Nice to meet you.
Well,
Yeah.
it's not an accident.
Right.
so, Tyler, just speaking with, he told me that, yeah, you're set for lunch in 176 Hudson, and he'll make sure that you're introduced, get to meet the grad students.
I can handle
It's a
that too.
customary thing, so that's great. You're
Yeah.
willing to go.
Just gonna put the room number down here because my brain will empty out as soon as I give my talk. Guess I could wear my watch instead of using my phone as the.
How did we get you? I was almost
I almost never. I mean, it takes me months to get through my email, so I appreciate you. Yeah. And they like invasion. So. Hey, lose the...
It's just different. Good.
How are you? No, I'm not meeting with anyone that I have.
Pretty good.
Well, I'm going to come in around lunch. I might be a meeting after that, but I'm literally, the meeting is, I'm missing this meeting. We're on the 27th, so it's probably maybe I could be around for the last. I'll come and grab you. Okay. I know where they use that.
Well, good morning, everyone. It's 9 o'clock, so let's start. Good morning, Dick. Well, this is not a job interview seminar. You might wish it was. So we're going to have one more job interview seminar next Monday. But today, we begin our official winter quarter PLP 290 seminar series. We're lucky to have with us this morning Dr. Jonathan Eisen. He's professor, Department of Evolution and Ecology, and also in the Department of Medical Microbiology and Immunology. And he's got appointments with the UC Davis Genome Center, and he's an adjunct scientist with JGI. His education, he received his AB degree from Harvard in 1990, and his PhD from Stanford University, 1998. From 2000 to 2005, he worked in the Department of Microbial Genetics at the Institute of Genomic Research, Rockville, Maryland. And then he came to Davis right after that. His current research is focused on in the area of microbial genomics and microbial community analysis, with emphasis on evolution of functions, processes. He's a very active scientific blogger. You can read his blog online. He's a proponent of open access scientific publishing. He's received several awards, Best and Brightest Innovators, Benjamin Franklin Award for contributions to open science, and he's a fellow of the American Academy of Microbiology. And I listened to his TED Med Talk from last year online. You can find out more about Dr. Eisen in that talk. So thank you very much for speaking with us. And his title is Phylogeny-driven Approaches to Genomics and Metagenomics. Dr. Eisen.
Thank you. You didn't mention one thing. This is my Twitter handle. I'm more obsessed with Twitter now than blogging, but we can talk. I can talk to people about that afterwards. So the title is accurate in what I want to talk about, but really what I want to talk about is a derivative of this title, which is really the importance of history. And that's where phylogeny-driven approaches come in. And I'm not just going to talk about using phylogeny to study organisms, but I'm also going to talk about my own personal history in leading up to where I am, as well as the history of the field of DNA sequencing, or the history of the use of sequencing approaches. And I'm going to try and weave these three things together. There probably will be some broken strands in this weaving, but bear with me. So I believe that there are sort of four major eras in terms of DNA sequencing to study microbes. And the first era of this is studies of microbes to build a tree of life, a full tree of life. And this really, in terms of sequencing, began with Carl Woese and others. And what I want to tell you about is how this has had a big impact on me and my personal history, but also many other people in fields as a sort of model here. And so I heard about Woese's work. I even worked as an undergraduate on microbial diversity a little bit, but I went to graduate school to work on butterfly population genetics and evolution. That didn't work out. And I switched to another lab that wasn't working on either of the things I thought I was interested in. It was a lab studying DNA repair processes. And I was a little lost, shall we say. And I felt like I needed some sort of guidance roadmap to get me going. And my roadmap was this, was a paper by Carl Woese from 1987 that I carried around with me everywhere that was a compendium of everything they knew at the time on the phylogeny and evolution of mostly bacteria and archaea, or archaebacteria as they used to be called. And I sort of had this in my head over and over and over again, and I decided that rather than focus on some particular molecular or mechanistic detail of DNA repair processes, as most other people in the lab worked on, I was going to use this map as a guide to find things that other people weren't working on. And what I basically did was I surveyed PubMed and basically the field and discovered that almost all of the experimental studies of DNA repair, recombination, and replication processes, the RRR field, were in these 4 groups of organisms. Purple bacteria, also known now as proteobacteria, mostly E. coli, animals, mostly mouse and humans, fungi, mostly Saccharomyces cerevisiae, and a few studies in plants. And there was a very limited sampling of the rest of diversity of life. And so my focus for my PhD research was basically to sample across the diversity of life in terms of various aspects of DNA repair processes. And I went after a diversity of organisms to do both cloning studies and experimental studies, and I also started to do bioinformatics to analyze what was known about DNA repair processes and their genes to study their evolution of them. And I spent a lot of time, for example, focusing on halophilic archaea, these extremely salt-tolerant archaea that also happen to be highly radiation resistant, trying to figure out what their mechanism of radiation resistance was in comparison, for example, to E. coli. And there was basically nothing known about archaea DNA repair processes at the time, so I did some of the sort of traditional experimental studies of DNA repair processes in this organism. I chose it because it was one of the few archaea at the time that had any genetic tools available. You could do a few targeted disruptions. There was a physical map and a genetic map and so on. And it was and still is a model organism for studying archaea. I also became a bit obsessed with evolutionary studies of one particular DNA repair gene, REC-A. The reason I focused on REC-A was many people were going after REC-A to sequence and then make knockouts of, because REC-A regulates homologous recombination across the diversity of life, or homologs of REC-A. And if you want to have a recombination minus strain, which is very useful for many purposes, you needed to clone the REC-A and then knock out the REC-A. And so after ribosomal RNA sequences, the next most diversely sequenced gene in bacteria was REC-A. And so I did a lot of work on comparing the structure of phylogenetic trees between REC-A and ribosomal RNA, and basically showing that they were, for most purposes, virtually identical to each other, which suggested that either both were biased in the same way, or that they were really reflecting the true history of organisms from which they came. And along the way, although from my personal timeline, this is in a different order, after Woese and other people started to show that you could build a tree of life based upon sequences, the next era to me in terms of sequence-based studies of microbes is going out into the environment, collecting samples without growing them in the lab, and sequencing ribosomal RNA genes, work pioneered by Norm Pace and colleagues. You go out to the environment, now with PCR, but before it without PCR, you sequence ribosomal RNA genes, and you build an evolutionary tree, and it can tell you something about the organisms that you've never grown in the lab by comparison to organisms that have been grown. Most of the work on environmental DNA sequencing for many years focused on ribosomal RNA because they're really good PCR primers that you can use because the sequence is conserved across the diversity of life in certain regions that sets you up for good PCR. But this created some sort of fretting in the community, because what if the ribosomal RNA sampling that we were doing was not telling us everything about the diversity of life? And I spent a lot of time now, after doing my evolutionary studies of REC-A, trying to, just as a personal history, before I went to graduate school, I actually worked on using ribosomal RNA PCR to characterize uncultured microbial symbionts that live inside invertebrates and serve as chemosynthetic symbionts for these invertebrates, and basically doing the same thing that most people were doing in ribosomal RNA, building an evolutionary tree and trying to figure out what these uncultured organisms were by placing them in a phylogenetic tree. So what I really wanted to do was take what people were doing with ribosomal RNA, which was going to environments not just where there was one organism like the symbionts, but two organisms, and you could say something about the diversity of those two organisms, or four organisms. You could say something about the diversity of the four organisms, and so on in environmental communities. And I really wanted to try to do this with REC-A, and I spent a lot of time designing degenerate PCR primers based upon the conserved amino acid sequences of REC-A to try and use this from environmental samples. It worked okay. Mostly from cultured organisms we could get it to work. It didn't work quite as well from environmental samples, but I actually had a postdoc lined up at Berkeley to go work on REC-A cloning from environmental samples with the person who discovered REC-A, John Clark, and Norm Pace, who had moved to Berkeley and was also interested in REC-A from environmental samples. But REC-A from environmental samples didn't really work well, and I was learning this fortunately before I started that postdoc. And I'll come back to that in a couple of minutes. So the field has been dominated by people doing ribosomal RNA PCR because the primers work really, really well, and they just don't work well for even highly conserved protein coding genes across the diversity of life. And so I've spent a lot of time now using ribosomal RNA PCR in my environmental studies, like many other microbial ecologists. And the main reason that many of us are still or are doing lots of ribosomal RNA PCR and sequencing is that sequencing now has gone crazy. Sequencing has gotten incredibly cheap and easy and efficient. And whereas when I was in Colleen Kavanaugh's lab studying these chemosynthetic symbionts, it took me a year and a half to sequence one 16S ribosomal RNA gene, and I got a paper out of it. Now we can do 100 million 16S ribosomal RNA sequences in a MySeq run for $1,000. And so the field has just exploded in terms of what you can do with this. You can generate, for example, more PCR products, so you can sequence deeper into a community if there was a lot of diversity there. And that deeper sequencing allows you to sample not only rare organisms, but get a better picture of the relative abundance of organisms that are in that sample by counting the number of hits that you get to different taxa. And of course, one thing that has come up in the last couple of years has been that you can use this massive amount of sequencing to characterize many, many, many samples at once by basically adding little sequence-based tags to each sample and then pooling all the samples together into one run of one of these sequencing machines, deconvoluting the data afterwards, and that allows you to do time series, fine-scale environmental sampling, lots of experimental studies, and so on with ribosomal RNA PCR for not too expensive, not much expense. And in my lab, we have basically been, I would say, co-evolving with the ribosomal RNA sequencing community, trying to figure out what we can use ribosomal RNA sequencing to study the ecosystems that we're interested in, or the fundamental ecological and evolutionary questions that we're interested in. We've collaborated, for example, with Jen Martini on looking at biogeographical patterns of microbial diversity and trying to compare those biogeographical patterns to what you see in plants and animals. By sequencing being cheap, this is the first time that you can really do this globally across microorganisms in comparison to plants and animals. I've never lost this, what was ingrained in me in graduate school, this desire to work with model organisms. And so when I moved to UC Davis, one of the things that I really wanted to do was collaborate with the people who were experts in Drosophila in particular and start to work on the microbial diversity in the Drosophila gut. And we've collaborated with Artyom Kopp's lab, and a graduate student who was in his lab, Angus Chandler, and Jenna Lang, who was a graduate student in my lab, worked with them to start to look at the microbial diversity in the Drosophila gut as a model system in parallel with all the work that's being done in the human microbiome and the mouse microbiome and the fish microbiome, Drosophila turns out to be a really good model, in part because you have the ability to do large numbers of individuals and nice genetics in the host that you can now layer on top of that studies of the microbes that live in and on them. I'm not going to go into all the details of these various studies, but basically, since I came to UC Davis, one of my main missions was to help other people on campus work on microbial diversity in their system and teach them about the tools that were available and help them with the bioinformatics. Initially, this came from my lab, so we've collaborated again with Artyom Kopp's lab and with many other labs, including Jay Solnick's lab in MedMicro, studies with Miriam Martin and a grad student in my lab, Srijak Bhatnagar, who were looking at the microbial diversity associated with Helicobacter pylori infections in macaques. The details, again, I don't think are what I want to emphasize here, but what I want to emphasize is that I think that just about anybody studying a model plant or animal might be interested in looking at the microbial diversity associated with that plant or animal under certain conditions. Not all conditions, but under certain conditions. And it's not that hard to do this anymore. And we got overwhelmed with trying to do this in my lab. And fortunately, I convinced what used to be the microarray core in the Genome Center. In case people don't know, most people don't do microarrays anymore, or not as many. They are now the host microbe systems biology core, and they provide as a service ribosomal RNA sequencing and some help with the informatics for anybody on campus to analyze the microbial diversity associated with their sample for sort of a recharge. The fact that sequencing has gotten so cheap has expanded where people are looking at microbial diversity, not just in the human gut or in soils or in places you might have imagined people studying microbial diversity, but now there are a lot of people that are exploring the microbial diversity, for example, of the buildings that are around us. There's a Sloan Foundation program that we have a grant from called the Microbiology of the Built Environment Program. There are about 20 labs that are funded to do some type of microbial ecological study, linking that study to hopefully building science parameters about some built environment facility. It's even gotten to the point, and I was just listening to this really interesting thing on NPR this morning about in the Central Valley in California, they're trying to get farmers around the Central Valley to keep their rice fields flooded for longer in order to help bird populations, especially in this drought time. And one of the things they're leveraging for this is a tool called eBird, which is an iPhone app. Oh, I put away my phone, but I have it on my app, where people can record sightings of different birds and their GPS coordinates wherever they are. And the Cornell Bird Observatory and others have used this information to create almost a real-time global map of bird diversity around the country. And you can respond to this global map in very dynamic ways. What we would like to do is the same thing for microbial diversity. We would like to engage the public in sampling microbial diversity in the same way that people are engaged in sampling bird diversity. Now, it's a little bit harder. You can just go out with your binoculars and see the birds. You can't quite do the same with most microbial diversity, but we're working with a variety of groups trying to design methods for people to go out and collect swabs and then get back culture and or sequence data relatively rapidly and create a global map of microbial diversity with a citizen science project. We're focusing initially mostly on just sort of simple surfaces like phones, but you could do this with anything. There's even crowdsourced activities related to the microbiome now. So the microbial diversity is kind of a fad, but for good and bad reasons. I don't want to belabor the point, but there's just an absolutely massively growing arena of people using ribosomal RNA PCR to characterize microbial diversity in a particular sample. And what we focus on mostly in my lab is not particular sites or samples, but what we really focus on is the tools by which people can analyze that data that is coming out, large piles of sequence data, and how do you analyze it. And the tools that we work on are those driven by phylogenetic comparisons. I've always been sort of obsessed with this, the historical perspective on biological diversity. And what we do is try and add a layer onto tools that people use to study ribosomal RNA sequencing by developing high throughput phylogenetic approaches to doing this. And phylogeny is not, it's not me, I have not invented the use of phylogeny in studying ribosomal RNA. This has been there from the beginning of the field. People used phylogeny as the way of saying, I have a new sequence, where does it sit relative to other sequences? But you can also use phylogeny to do things like look at rates of change among taxa. You can use phylogenetic diversity metrics to study the ecology of a community. There's a lot of work, people may be familiar with the tool Unifrac, which is basically a phylogeny-driven method to compare the diversity between two different microbial communities. And what we've been trying to do is layer onto what people are doing other tools to use phylogeny. So a few years ago, we built what was really the first completely automated pipeline for taking large amounts of sequence data, ribosomal RNA data, and having as an output phylogenetic trees and taxonomic assignments of those organisms based purely on the phylogeny, rather than what most people were doing once they started to generate lots of sequences was to do a blast search, a similarity search with their sequence, and pull up the best hit in the blast search and say, most likely my organism is this. But similarity scores are not the right way to assign taxonomy to organisms or environmental sequences. A phylogeny is better, and we've been building tools that can do this, in essence, in a high-throughput, automated manner. A graduate student, Amber Hartman, in my lab, really didn't like, I mean, she was involved in this study, this STAT pipeline, but she didn't feel it was comprehensive enough or user-friendly enough. And so she worked with a couple of people here in computer science, including Bertram Ludescher's lab, to develop a completely automated workflow that would take you from your raw data to even producing figures at the end for comparative analysis and building trees and making principal coordinate plots. And it's all done within this open source scientific workflow package. So we've been working on these sort of automated methods to analyze the data and also trying to figure out how we can analyze the data better. And so I've been in this collaboration with Katie Pollard and Jessica Green's lab for a few years, funded by the Moore Foundation. And one of the postdocs on the project, Stephen Kemble, developed a really nice tool for doing something that's really important in ribosomal RNA studies, which is correcting for the copy number differences between organisms. So a lot of people would like to take ribosomal RNA data from a community and count the number of ribosomal RNA sequences that you get out after PCR and use that to infer information about the relative abundance of the taxa in the environment. Unfortunately, the copy number of ribosomal RNA genes varies a lot between taxa. And so if your taxa has 10 copies of the ribosomal RNA, you might overestimate its relative abundance by counting ribosomal RNA genes compared to an organism that has 1 copy of the ribosomal RNA gene. And so he built a phylogenetic tool that will, in essence, integrate the phylogenetic tree of taxa with known copy number data from genome sequencing projects largely, and estimate the copy number for individual sequences that you have from an environmental sample and correct for the estimate of their copy number to your relative abundance estimate. Tom Sharpton, who was also involved in this project, was a postdoc in Katie Pollard's lab. He built a tool. So many people may be familiar. You go to an environment, you sequence ribosomal RNA genes, and one of the things that people really want to do is cluster them into what are called OTUs, operational taxonomic units. That is, sequences that are really similar to each other from your sample, so similar that we might call them the same species, for example, or because we don't want to use the term species, we call them OTUs, or operational taxonomic units. And that collapsing into clusters is largely done by non-phylogenetic methods, by similarity-based methods. And those have certain areas where they perform poorly. So Tom developed a phylogeny-driven method to do this OTU clustering rather than a simple similarity-based metric. And my lab still works on this ribosomal RNA sequencing tools because ribosomal RNA sequencing is not going away and is very powerful and useful. And I'd be happy to talk to anybody if they want insights or advice or complaints about ribosomal RNA tools that are out there. But clearly, ribosomal RNA sampling from communities and from microbes is not enough. There's a lot missing when you do just a tree based upon a ribosomal RNA or a tree from an uncultured organism based upon a ribosomal RNA. And in terms of sequencing and the history of sequencing, the next era was taking organisms that you could grow in the lab and sequencing their genomes, their complete genomes. So rather than sequencing just 1 or 2 genes from the organism, you could now sequence their complete genome and make many hypotheses or learn many things about the organism. This doesn't, of course, obviate the need to do experimental studies, but I'm focusing on the sequence-based approaches here. And so this was launched in 1995 by the Institute for Genomic Research, where Craig Venter and Ham Smith got together and were the first to use the random shotgun sequencing method to sequence a large genome, a 1 million base pair genome from this bacteria. Fred Sanger actually had used this random sequencing approach previously to sequence phage genomes, but nobody had done it with bacterial genomes until they did it in 1995. So back to me, I was in graduate school when this happened. Ham Smith came and gave a talk at Stanford right when I think the first paper was about to come out, and he showed a table in his talk of all the organisms that Tiger was sequencing the genomes of. And every organism that I was trying to clone out DNA repair genes by degenerate PCR from, struggling to grow these organisms and get degenerate PCR to work, was on his list. I was screwed. And, well, I mean, really, it was like, they're going to sequence the whole genomes. Why am I toiling away sequencing individual DNA repair genes? So the first thing I did was, if you can't beat them, critique them. And so I shifted a lot of the work I was doing from laboratory work to more bioinformatics. And one of the things that I was doing a lot of was building evolutionary trees of DNA repair genes and trying to predict functions of genes that had never been experimentally characterized by these trees. And I'd written a couple of papers on this, but Rick Myers and Dale Kaiser got asked to write a commentary on this genome that had come out in 1997 from Tiger. And they had heard me ranting and ranting about all the flaws of genome bioinformatics methods for years. And they said, basically, put up or shut up. Find something wrong with this genome. And amazingly, I went and read the paper on this genome, and literally the first gene that I looked at, they had done something strange with, and it was a DNA repair gene. So what I looked at was this quote from the paper: the ability of H. pylori to perform mismatch repair, which was one of the processes I was studying. That's the process by which organisms scan newly replicated DNA to look for mismatches between the old DNA strand and the newly synthesized DNA strand. And they will remove a whole region of the genome on the newly replicated strand if there's a mismatch, which implies that there was a replication error. So again, they said, the ability of H. pylori to perform mismatch repair is suggested by the presence of methyltransferases, which are used in E. coli for this, MutS, which is one of the genes in E. coli used for mismatch repair, and UVRD. However, orthologs of MutH, another gene, and MutL were not identified. Now, this immediately struck me as strange because I had spent 3 years trying to clone out MutS and MutL homologs from across the diversity of life, because experimental studies had shown across every organism that had been found to have mismatch repair, biochemically, genetically, or otherwise, that it always required homologs of MutS, which recognizes the mismatch, and homologs of MutL, which the exact function was unknown, but every organism that had mismatch repair had both of these. No organism previously had had one, but not the other. And so I was a little skeptical of this assignment of mismatch repair to this organism based on the presence of a homolog of just one of the 2 genes known to be essential for mismatch repair. So I went in and looked in the detail at what they did. They did a blast search. Their blast search with their gene pulled up sequences that in the database had been annotated as DNA mismatch repair protein, yada, yada, yada. And they inferred that their gene was a mismatch repair protein and the organism had mismatch repair. So, make a very long story short, I built evolutionary trees of all the MutS homologs that were available at the time, overlaid onto those trees known experimentally determined functions. In red are the functions that the genes that were known to be involved in mismatch repair. You can't really see it that well, but in blue over here are genes that were not known to be involved in mismatch repair. They had other functions. And the Helicobacter pylori gene is over here, in a subgroup for which no one has experimentally determined any function at the time. And it wasn't particularly closely related to any genes that were known to be involved in mismatch repair. So I predicted in my paper that this Helicobacter pylori would be unlikely to have mismatch repair. It does appear that that is correct. The mutation rate is quite high in Helicobacter pylori, and it appears that it does not have normal post-replication mismatch repair. So I invented a name for this. Editors of one of the journals made me invent a name for it. I called it Phylogeny-driven. I now rant and rave against new omics terms, but this is what it is. Basically for the need for integrating genome sequence analysis and phylogenetic analysis in order to make sense out of the new genome data that was coming out, which, genome data was being streamed out without a lot of functional studies associated with it. And basically I argued that you could use character state reconstruction methods, which are a standard tool in phylogeny, to take an evolutionary tree of a protein family. And like I did with MutS, overlay onto that evolutionary tree known experimentally determined functions, not predictions of functions, not names from the database, but experimentally determined functions. And then based upon where your sequence of interest sits relative to known functions in the history of the evolution of function, you could predict the function of those genes by that phylogeny. Now, this may sound somewhat novel. I think it was a little bit novel, but really I literally stole what everybody was doing in ribosomal RNA studies for organisms and applied it to proteins. What people were doing with organisms was sequencing from environmental samples, building an evolutionary tree compared to known organisms, placing their unknown organism in the evolutionary tree relative to known organisms, and then trying to predict the functions of the unknown organism based upon the functions of the other taxa that sat near it in the evolutionary tree. This is the exact same concept, but now to protein families instead of organisms. This turns out to be a very powerful tool in the prediction of function of uncharacterized genes. Lots of people, myself included, but many others have developed this further, much better than we do in terms of computer science, for automated methods for doing this phylogeny-based prediction of function. And I'll come back to that in a minute, but so here I am critiquing Tiger for their approach to annotating genomes and pointing out that they're making mistakes by relying on blast searches. And amazingly, I finagled my way into a dinner with Craig Venter when I was at the time fretting about my upcoming postdoc that I had signed up to do that seemed to be impossible on Rec-A and environmental samples. And I'm at this dinner with Craig where apparently he was planning Celera with some big shots from Silicon Valley, and somehow I had gotten to this dinner. And he hears I'm an evolutionary biologist. He starts telling me about something they were doing in evolution. And I start drawing on a napkin proving to him why he was wrong. And by the end of the day, I had a job as a faculty member at Tiger. And eventually my wife, who was working at a plant genomics company after her graduate work, got a faculty job at Tiger too. So we ditched the postdoc and moved to Maryland, and I went to Tiger, and both me and my wife got faculty jobs at Tiger. Now, it's probably better for me that right after I accepted the job, Craig announced he was leaving Tiger to go start Celera. So actually, I never worked for Craig, which I hear is not always fun. I worked with him, which was great, but working for him is a little bit different. And I still work with him, which I like quite a lot. Claire Fraser, his then wife, became the president of Tiger. And I basically spent 8 years as sort of the resident evolutionary biologist at Tiger. Whenever there was some organism that was, thought to be weird, that is evolutionarily interesting, people would send it to me, or whenever they needed help with some type of evolutionary analysis with their genome, they would come to me. And it was like a kid in a candy shop for 8 years there. I mean, they were sequencing, the genome, the first plant genome, the first malaria genome, the first streptococcus genome, all these amazing first sampling of genome sequences. There was always something interesting to do evolutionarily with these genomes. I got to run eventually my own genome projects on weird organisms like Tetrahymena and Wolbachia and weird symbionts that nobody else at the Institute cared at all about. But basically, I spent a lot of time working on what I call phylogenomic methods development. How can you do comparative genomics and add a phylogeny layer to those comparisons? How can we predict which genes are involved in, which genes might be good vaccine targets because of their evolutionary rate variation, for example? that is suggesting they're interacting with a host immune system. And I'm not going to go into a lot of detail on those methods, but I spent a lot of time working on evolutionary phylogeny-driven tools to analyze genome sequences in much the same way that I've been working on phylogeny-driven tools to analyze ribosomal RNA sequences. And I'm just going to tell you an example of one of them. So if we go back to this phylogenetic tree-based prediction of function, there's one case, or unfortunately thousands and thousands of cases, but one situation where this doesn't work very well. So if you have a gene, and you do a database search with your gene of interest, and you find lots of homologs of your gene of interest. If some of those homologs have been experimentally characterized, then I would argue that you should build an evolutionary tree of those genes to predict the function. But in many cases, none of the genes that your gene is similar to have ever been characterized genetically, biochemically, or in any experimental way. So now you have a gene with lots of homologs. You could build every evolutionary tree you want, and it's not going to help you predict the function any more than rolling dice will help you predict the function. And so I spent a lot of time at Tiger trying to work on methods that could predict the function even in these cases. And what we did was basically co-opt methods that others had developed for something called non-homology-based functional prediction, originally developed by people like Eugene Koonin and Ed Marcotte and people in David Eisenberg's lab. These are ways to try and predict functions of genes without any of the gene or any of its homologs being experimentally characterized in a direct way. So a classic example of this is clustering gene expression patterns, looking for correlated gene expression of genes to genes that have known experimentally determined function, and an unknown, if it always has a correlated gene expression pattern to cell cycle genes, for example, you might predict that your gene is involved in the cell cycle. So the tool that we focused on in particular, I'll show you in 1 second, but the organism that we did this with is called Carboxytothermus hydrogenoformans, thermophilic bacteria that was isolated from a Kamchatka hot spring. It's related to, it's in the Firmicutes phylum, that is, it's related to Streptococcus, Staphylococcus, Bacillus, etc. And I don't want to go into all the details, but we noticed something interesting when we first annotated the genome, which was that there were a lot of homologs of sporulation genes in the genome. This organism was related to organisms that sporulated, but people had tried for 5 years to get it to sporulate and never found it could sporulate. After noticing the genes in the genome, our collaborator went back and tried a little bit harder and managed to get it to form endospores, just like you see spores in Bacillus anthracis and other Bacillus and Clostridium species. And then what we did was we took this method called phylogenetic profiling, where you group genes by their distribution patterns across species, not by their sequence similarity to anything. And we did this for Carboxytothermus hydrogenoformans, and we found a cluster of genes found in sporulating species and not non-sporulating species, of which many of these genes were annotated by us as being involved in sporulation. But many of the genes were annotated as hypothetical protein or conserved hypothetical protein. But they were basically found in this, all of them were found in the same set of species that all the sporulation genes were found. This included many genes in Bacillus subtilis that had never been experimentally characterized. So we predicted that these genes would be involved, likely be new sporulation genes that had previously been undiscovered. Rich Lozik's lab followed this up with a couple of papers, and we just had yet another paper basically that has shown that, in essence, every single one of these genes that was annotated as a conserved hypothetical protein but had a correlated distribution pattern to genes involved in sporulation are involved in sporulation. So this phylogenetic profiling is a really powerful tool for predicting function for genes that have not been experimentally characterized. And if you're analyzing genomes and looking for new functions to look at, I think this is the tool to use these days for comparative genomic analysis. So the last thing I want to talk about is the last era in sequencing and microbial diversity, which is to go the next step in terms of genomes and go back to environmental samples and start to characterize the genomes of organisms rather than just ribosomal RNA sequences from environmental samples. And it works in basically the same model as environmental ribosomal RNA studies, except there's no PCR. You pull out the total DNA from an environmental sample and you sequence it largely with random shotgun sequencing, and you generate short sequence reads from your environmental community, and then you freak out because the analyzing this data is just horrendous, mostly. Although it can get better, and what we're trying to do in my lab is develop the methods to try and make it work better. And again, shocking, largely what we've been doing is developing phylogeny-driven methods to analyze this type of environmental genomic data. You can do the same types of things that you would do with ribosomal RNA, but now you can do it with every gene across a genome, and you can even apply those tools to studying function, not just taxonomic diversity. The first two studies of this were in 2004, one from Jill Banfield's lab, one from JCV, the Craig Venter Institute, of which I was a collaborator on, going out to environmental samples. And what we showed in the Venter paper was basically you can do the same things that people were doing with ribosomal RNA to look at taxonomic diversity or richness, but now with a diversity of other sequences, with protein coding sequences. So not just ribosomal RNA, but you could finally do this with Rec-A. I was joyous. And with lots of other genes. And it turns out that it actually works better with protein coding genes than with ribosomal RNA, probably because of the copy number variation in ribosomal RNA. And many protein coding genes are single copy across a large diversity of organisms. We developed high throughput tools for doing this, much like we developed with ribosomal RNA. You can predict function with phylogenetic trees from environmental data, just like you can from cultured organisms. We use phylogenetic trees to sort through sequences to try and say what organism they came from. This is something called binning. It's probably the biggest challenge in environmental studies, and phylogenetic trees can help with that. And then what I want to spend the last few minutes on is talking about what we're doing now with metagenomics, which is really trying to think about that, screen picture and how we can improve our ability to analyze metagenomic data, largely by, again, using phylogeny-driven tools. And I'm going to give you 3 examples of this, basically. So in the first example, we took a step back and said, okay, we're going to go out to these environments and do environmental sequencing and try and analyze the data that's coming back. That's really hard for many reasons, but one reason it's particularly hard is the sampling that we have of reference genomes to compare everything to. So if you go to the evolutionary tree that was available, many years ago and ask how are the genomes being sampled, they're incredibly biased across the tree of life. We got a grant when I was at Tiger to fill in what we thought was filling in the bacterial branch of the tree of life by doing a genome from each of the 8 phyla of bacteria for which there were cultured species and no genomes available. And we were proud of this project, sequencing the first representative of these 8 phyla, and we realized halfway through the project that this was nowhere near enough sampling of diversity of life. One genome for a phylum that is probably 2.5 billion years of evolution is not particularly useful. So when I moved here, I had an adjunct appointment at the Joint Genome Institute, and we, with many people there, developed this project called the Genome Encyclopedia of Bacteria and Archaea, marching our way through the phylogenetic tree of bacteria and archaea, sequencing genomes of cultured organisms to really fill in the diversity of bacteria and archaea across the tree. We showed basically that this is a really good tool for discovering protein family diversity. If you sequence phylogenetically novel organisms, you're more likely to find a new protein family than if you sequence non-phylogenetically novel organisms. Found all sorts of weird things in some of the genomes, including eukaryotic-like actin in one of the bacteria. And this spawned many what we call GBA side projects. There was a project led by Cheryl Kerfeld to fill in the cyanobacterial diversity using a phylogeny-driven approach. We've been doing a project here with Mark Facciati to fill in the halophilic archaeal diversity for the phylogeny-driven approach. And what this was missing was basically, this helped a lot for analyzing metagenomic data, but what we and other people showed was this: we were basically sampling like 1/10 of 1/8 of 1/10% of the true diversity of bacteria and archaea. And to get at the true diversity, you have to go at uncultured organisms. That's where the diversity lies. And so Tanya Wojcicki at the JGI led a project that they call the dark matter of biology, which was now marching through the true tree, including uncultured organisms, and using single-cell genomics, where you take a single cell and amplify the genome and sequence the genome to fill in the genomic data from across this diversity of life. We just collaborated with her on a paper that came out a few months ago with the first 200 of these genomes. They're doing another couple of hundred. It's still not delving deeply into the phylogenetic diversity of bacteria and archaea, but it is starting to make a dent. People need to sample the other microbes, viruses, and eukaryotes, but I haven't convinced anybody to fund that yet. We're working on it. The second thing that we've been trying, so sampling diversity is critical for making sense out of any of this environmental data. The second thing we've been trying to do is develop methods that take advantage of diverse genome sequence data and adapt our methods that we were using for analyzing metagenomic data. And I've sort of hinted at this before. We had a grant with Katie Pollard, who used to be at Davis and is now at UCSF, and Jessica Green, who's in Oregon, to develop these tools funded by the Moore Foundation. And I'm just going to take you through a couple of these tools. One of them was you can't do anything by hand anymore with this amount of sequence data. One thing that you like to do with sequence data is throw away regions of sequence alignments that are ambiguous or misleading before you build an evolutionary tree. So Martin Wu, when he was in my lab, developed a tool that will do this in a completely automated manner to do this masking of protein family sequence alignments, called it Zorro. And that helps you do high throughput automated phylogenetic trees of any protein family. Stephen Kemble developed another tool which was to do phylogeny-based studies of environmental data, but now combining sequences of different genes into one giant phylogeny of the environmental sample, in essence. We've been collaborating with a couple of mathematicians to try and figure out how to do the phylogenetic profiling equivalent with metagenomic profiling, with grouping sequences from environmental samples and trying to see what their co-occurrence patterns are and whether or not those co-occurrence patterns are related to any environmental data like temperature, et cetera. This turns out to be very tricky, but we are making some progress on that sort of environmental clustering. We've also been developing high throughput tools like STAP and Waters, but to do automated phylogeny and automated analysis of protein family sequences from metagenomic data rather than just ribosome RNA. We had one tool that a paper came out last week called PhyloSift, and this is basically you plug in your environmental data and it will build phylogenetic trees of all gene families in that data set, in particular ones that are known good phylogenetic markers, and give you an output of taxonomic and ecological diversity metrics. And then the last thing that we're working on is to try and improve the data sets that we have available to make sense out of environmental data. And in particular, what we're trying to do with this is build pre-computed alignments and phylogenetic trees and sorting of known protein family diversity in genomes so that we can then go to environments and in a high throughput manner analyze those protein families without lots of manual curation. So we're doing this to develop alternative phylogenetic markers for different taxonomic groups to go with REC-A and ribosomal RNA and other known phylogenetic markers. We just published a paper on doing this where, for example, for some groups like cyanobacteria right now, there are 590 proteins found across cyanobacterial genomes that seem to be robust markers for doing the phylogeny of cyanobacteria. So if you have an environment with cyanobacteria in them, you probably can, almost every fourth gene that you hit is a good phylogenetic marker for a cyanobacteria. And basically, we're developing the tools such that we can analyze massive data sets when they come out. And then the last thing we're doing is, to place this all into context, you need a tree of all the genomes that are out there. Turns out that with 4,000, 5,000 genomes, it's getting hard to build a phylogenetic tree of all of those genomes. And Jenna Lang, this was a grad student in my lab and is now a postdoc, has been trying to develop ways to build automatic manner trees of thousands and thousands of genomes so that we can then anchor the environmental data. And so I'll just end with, I mentioned eBird. That's not an accident. I've been an obsessed birder when I was a child. My first research was on hummingbird physiology, and I've always carried bird books with me wherever I go. I used to give them out as gifts to everybody, a little strange. But What I want to do, field guides are amazing. They tell you where organisms should be. They tell you the taxonomic diversity of organisms, the ecological diversity, the functional diversity. And you can use them to interpret if you make an observation and say whether or not it's unusual or not. What I want is the same thing for microbes. And that's why I've been pushing into the use of sequence data in particular. It's not that sequence data is the only way to study microbial diversity. It certainly is not. But because it is so cheap now, and so easy now, and you're able to do this on a massive scale, it would allow us to potentially start to create a field guide to the microbes like we have with a field guide to the birds. And I will end it there and thank all my millions of collaborators and funding agencies. Thank you.
Thanks, Jonathan. Time for a few questions. Yeah.
Can phylogenomics provide an answer for the fact, I guess, that there's very few pathogens among the archaea? Is there anything you can learn from their genome?
Well, I mean, there are many papers out there analyzing archaeal genomes to try and compare them to bacteria, and in particular bacterial pathogens, and to compare them to eukaryotic pathogens as well, and trying to understand what makes them different. There are some gene families that people have found that, or domains actually, that people have found that tend to not occur in the archaea. The main argument that I think has been made that is the most convincing has to do with membrane structure and sort of cell envelope structure. That is, they don't do a good job of living inside other organisms because of this. But there are a few archaea that can live inside at least other archaea. I don't know of too many that are intracellular symbionts of known other organisms, but that only explains the intracellular pathogens, right? There are plenty of extracellular bacterial pathogens, and there are no known archaea that have any known pathogenic effect. So I haven't seen anything convincing, but we haven't tried to do it. Maybe we'll try. Yeah.
So what about when you go into environmental samples and they're relatively complex, and so you have trouble trying to assemble whole genomes from them, and you have problems where you have a situation where the accessory genome is contributing in a significant way to adaptive processes, but its phylogenetic signal is inconsistent with the core genome. So what's the prospect for getting sort of high throughput assembly from these complex data sets?
So what we're doing right now, and what many other people are doing, is trying to leverage either pseudo or real long-read sequencing technology or paired-end technology to try and get that information. So for example, we're using this method from a company called Moleculo that was just recently bought by Illumina. And what they do is they take DNA from your sample, 10 to 20 kb pieces, and they basically, in a high throughput manner, take each individual fragment and make a sub-library of it. And then you tag that fragment. That fragment gets AAAA across all the sub-library. And fragment number 2 gets ATTT. And fragment number 3 gets CCCC. You pool it all together into Illumina, you sequence it, and then you deconvolute which sequence came from which fragment by those tags. And then you assemble them back together, and they call them pseudo-long reads. It's pretty nice. We just got. They won't tell anyone. We think it's with microfluidics, not emulsification, but they have not been very apparently clear about exactly what they're doing. But the data looks really nice, and we are using it to start to stitch together larger genomes from environmental samples. The other alternative is real long-read technology like Pacific Biosciences, which we're also trying to use, and the new technologies which are kind of pie in the sky but claim to be getting 100,000 base pair reads. So I think that those will allow a little bit better sorting and pairing of functional genes of interest and taxonomic marker genes. Yeah.
I spend a lot of time trying to identify fungi with their barcoding locus ITS.
Yeah.
And you think, well, man, it would be great if someone could automate this, but I have to read 2 papers per taxon that I try to identify. And I just, I know that it's like you just won a Grammy. I'm asking, well, what about the Oscar? But like, what can we do with eukaryotes? Can we do is there any chance of having an RDP for fungi anytime soon that actually works?
Yeah, so I mean, there are efforts with fungi to try, there have been historically efforts to get people together to build those data resources, and I know there's still ongoing conversations about that. Part of the problem has been that no one can keep up with any of these data sets, and so I don't know if I have a good model for even doing this, even maintaining the ribosomal database with bacteria, which was good. I mean, there are probably 10 trillion ribosomal RNA sequences for bacteria out there. And there are, 8,000 organisms that have a described species name, and they haven't even all had their ribosomal RNA sequence, let alone had functional studies go with them. So, I mean, I'd love to sit and talk about it, and we've been pitching this to various funding agencies, but I don't know how we will even keep up, even if it was fundable. So, but it would be good. I agree. Yeah.
What roles do genetic recombination and environmental selection play in phylogenomics?
So I skimmed over horizontal gene transfer, which is a big issue in any type of phylogeny-based study. And recombination within species is also a big issue within any phylogeny-driven approach. In general, my feeling is that phylogenetic approaches are the best approach to use whatever the history is. But you have to have a model that allows for reticulation in your trees or recombination within species in order for them to work well. So I dropped out the slides at the last minute. I had a bunch of slides on lateral gene transfer and recombination and how you can account for that in some of these analyses. It is really important. I'd be happy to talk about it, but the key point is the history matters. Figuring out the history is useful. Complicated histories make that harder, but it's still more useful than, say, a blast search.
Okay, well, thank you very much. There will be a lunch for our plant pathology graduate students.
Will you carry us today?
No, guys, would be better.
This is great, including one of my old students.
Oh, from the class? Which one?
The one that has the little tube with the...
Oh, yeah, awesome.
My graduate student.
I know. I've modified it.
Yeah. All right, I'll see you.
What's the name of
What?
what's the name of the company? The technology for
Molecular.
molecular.
And they got bought by Illumina.
Ah,
So it's now Illumina.
thank you. Would it be possible to come and talk to you?